Welcome to week 7 of Biology 1021. This week we're going to be dealing with cellular metabolism, also known as cellular respiration. There are some great videos in here because some of this stuff here is going to get a little complex. Okay, so how this is going to work is we're going to introduce everything, and then we're going to walk back on it and walk back on it again and walk back on it again. There's lots of videos here, so by the end of this you should be pretty good. What we're going to be dealing with there today is how we create energy. How do we store energy? And you guys know ATP already, adenosine triphosphate in its active form, okay? And that's what we're going to be dealing with there today. And, we're going to be talking about glucose, sugar source, the easiest source of food to metabolize and to extract energy from. And I want you to take notice there of these diagrams, the area. And you'll see here that in this, whatever's happening here, which we'll get into soon enough, this is called glycolysis. Very ancient process. It's been around. It's exhibited in many creatures that are older than us. And you'll see where this happens. This happens in the cytosol of the cell. Okay, the cytoplasm. And then this process moves on into the mitochondria. Now remember, these are the powerhouses of the cell. This is where the ATP is made. So you're going to see that we're going to be talking about processes there within the cytoplasm. We're going to be talking about processes there that happen within the mitochondria. And you'll see there that there's an inner membrane and an outer membrane. Okay? And this is what we call the space in between, and that will become important there later. So, to no further ado, I hope you guys are all doing well and that you guys have read the chapter, and if you haven't, make sure to read it after. Thank you. Let's go. So, metabolic processes. Metabolism there is just one of those all-encompassing words that talks about all the chemical reactions that are happening in the body. And there are two kinds, okay? There's anabolism, which is building, and catabolism, which is breaking down. Anabolism, you're taking smaller molecules and building bigger ones. Okay? Requires energy. Makes sense, right? It takes energy to build, to take smaller pieces and make bigger pieces. Catabolism, breaking down, is the opposite, where you're taking larger molecules and breaking them down into their smaller constituencies. This allows for the release of energy. This one requires energy. Catabolism releases energy. Okay? And you've seen this slide there before, the whole idea. Anabolism, building, requires ATP. And catabolism, you're breaking down these bigger molecules, you're releasing energy in the process. Okay? So, when we're talking about metabolic processes, you need to understand what an enzyme is. And an enzyme is a protein. We looked at the primary, secondary, tertiary, quaternary structure there of proteins there last week, right? And the way these amino acids come together, and depending upon their R groups being polar and non-polar, you're going to get these shapes. What were some of those shapes there? There was a beta sheet, there was an alpha helix. And within any given protein there, these things are so big, you could have multiple alpha helices, multiple beta sheets. For whatever reason, that enzyme has to have a particular shape, a three-dimensional conformation, okay? Because that is going to lock onto its target molecule. And for the enzyme in the mouth there called amylase, it has a three-dimensional conformation that exactly fits a starch molecule. Okay? And these enzymes here, they control the rate of metabolic reaction. And what they do is they don't necessarily speed up the reaction, they increase the amount of connections there that can happen. And the nice thing is about these enzymes there is that they're reusable. And so you'll see there that an enzyme, okay, and then here are your two substrate molecules, your two target molecules, and you see how the three-dimensional conformation upon the enzyme exactly fits these two? Exactly. It's called the lock and key, okay? And then what happens there, the enzyme does its job, whatever it is. You take these two products and you make one, and then the enzyme comes away completely unaffected. Okay, great movie there. Have a look at it, pause the video, and away you go. So, enzyme action, you'll see here, it's basically all about what we call a metabolic pathway. Series of enzyme-controlled reactions leading to formation of the product itself. Each new substrate is the product of the previous reaction. Well, you'll see there, substrate 1 is altered here by enzyme A to make substrate 2. Enzyme B takes that one and makes substrate 3, which is taken by enzyme C to make 4. And the last enzyme in this metabolic pathway takes substrate 4 and makes the product. Okay, so enzyme names commonly, they reflect the substrate, have the suffix ase, okay? Examples, sucrase, lactase, protease, lipase, okay? Very good. So, cofactors and coenzymes, okay? These cofactors here, they make some enzymes active. Non-protein components can be ions or coenzymes, okay? Which are organic molecules that act as cofactors. And these include vitamins. So these just, they alter the activity, they increase the activity there of enzymes, okay? Make them more efficient. So when you get into this metabolic pathway here, what we just saw, and a huge focus in this course and the next course is homeostasis. Now, you remember there that word there from week 1. Whatever's going on outside, a blizzard, a drought, you know, whatever type of environmental disaster is going on outside, our internal bodies are designed to maintain a normal body environment, 37 degrees, normal pH of 7.35, all of these things here. And the whole idea is that the internal environment of your body should not fluctuate very much, okay, and it can't. And it's all about homeostasis. We have turn-on mechanisms and we have turn-off mechanisms. And this is all done by homeostasis. And one of the ways homeostasis does it is something called a negative feedback. And I call it the teeter-totter, or the seesaw, okay? And where the whole idea here where you saw this already, substrate 1 to 2 to 3 to 4, all done by enzyme A, B, C, D, and D finally makes the end product that we are looking for. And as more product is made, all the product here that is made, it starts to interfere with the production over here. So then eventually, at some point in time, there's going to be so much product that this mechanism will be shut down, so therefore the entire mechanism shuts down. And then when we start to run out of product, there's not enough product there to interfere with this mechanism, so then this mechanism starts to turn on again. Kind of like a teeter-totter, right? When there's lots of this shuts off. When there's not a lot of this turns on. Okay? So energy for metabolic reactions here is the capacity. Energy is the capacity to change something. It is the ability to do work. Okay? Like once you've got a battery, okay, a battery has a positive end and a negative end. Once you have this, you are able to do work. Okay? Common forms of energy: heat, light, sound, electrical energy, mechanical energy, chemical energy, okay? Like the whole idea of a car hitting another car. It makes a heck of a sound. A lot of that energy in the car crash was converted to sound, Interesting, eh? Energy can be changed from one form to another, though through cellular respiration, energy is transferred from molecules to make it available for cellular use. Okay, a little bit about energy there. There's a good little question for you to have a look. Okay, quite interesting, that one there. Which has more calories? Exactly, but which one is healthier? Aha, interesting. Okay, release of chemical energy. Chemical energy is held in the bonds between the atoms of molecules. We know this already. And is released when the bonds are broken. Absolutely. Cells obtain energy by oxidizing, breaking bonds of glucose molecules and releasing energy. So that's how cells get energy, by oxidizing, breaking bonds of glucose. The resultant energy powers anabolic reactions, is stored as energy, ATP, is also released as heat. Okay? So, overview of cellular respiration. Here we have a beautiful ATP molecule. Okay? Adenosine triphosphate, which is the primary energy molecule of all of our cells. Okay? ATP is composed of adenine. There's your nucleotide base. Okay? Ribose. There's your sugar. And then we have phosphate. There's 3 phosphate groups, and this is the there's the energy right in here. This third phosphate is able to provide the energy. Okay, so this right here, ATP triphosphate, this is active and ready to go. Once you've utilized it and you've done your muscle action or lifted that bicep, one of these phosphates will go, and this will now become adenosine diphosphate, AD as in dog, phosphate. It'll lose one of those, and then you have to eat another banana in order to put the third phosphate on there to activate this again. So, as we just said, the whole idea there, ATP to ADP, okay? You can have a look at that and you'll see what goes on. There's your ATP, you do your work, one of the phosphates goes, now you have ADP, you need to eat another banana, and now it's active. Perfect. Energy can be released from molecules that have carbon-hydrogen bonds, such as methane, natural gas, glucose, fatty acids. Anything there that has that carbon-hydrogen bond, there's so much energy wrapped up in that, and that's how we make our car drive. We put gasoline in it. Gasoline, carbon and hydrogen. We burn that. The energy makes the pistons go. All right? So, oxidation is the loss of electrons. Reduction is the gaining of electrons. Very important. You'll also get that in chemistry as well. During cellular respiration, electrons and hydrogen ions are moved from one molecule to another. Remember, Leo the lion says grr. Loss of electrons is oxidation. Gain of electrons is reduction. Leo the lion says grr. If you have a better one, let me know. That's as good as we can come up with. So, overview of cellular respiration. 1 gram of carbohydrates gives us 4 calories. 1 gram of protein gives us 4 calories. 1 gram of fat gives us 9 calories. 1 gram of alcohol gives us 7 calories. There are about 3,500 calories in 1 pound of fat. 1 block of butter is 1 pound. So if you eat 1 pound of butter, you're eating about 3,500 calories. Oof. Ugh. Makes my skin crawl just thinking about that. Respiration. So you'll see here, What plants do is they take carbon dioxide, water, and light from the sun to make sugar and oxygen. Everything there that we need. And cellular respiration is basically going backwards, okay? Because we have to feed the trees there with carbon dioxide because that's what they breathe. So then, in order to burn this sugar, okay, there's your glucose plus oxygen produces CO2, water, and there's that sun's energy back that was input right in here, okay, when the plants were making their food. All right, so this is what we mean when we say cellular respiration, the act of breathing. What's the point at the cellular level, right? So, little overview, as we just saw, there's your glucose plus 6 oxygen molecules produces 6 carbon dioxide molecules, 6 water molecules, plus an ATP. Okay, perfect. Well, it's a lot more ATP than that. We're going to get into that. You're going to see there that if you don't have oxygen, your amount of ATP is limited. If you do have oxygen, you got so much ATP. We'll show you this right now. So, anaerobic catabolism, okay, oxygen is absent, okay, anaerobic catabolism, okay, this is the thing here, there is no oxygen, and the byproduct there, lactic acid. Think 100-meter sprinter, okay, that person there is not going into oxygen debt, they're running 100 meters, solely based on the oxygen that's in their myoglobin and their muscles already stored and the sugar ready to go. Okay, you do not get a lot of ATP production from this, but you do get some. Byproduct is lactic acid. That's the pain and suffering after doing an anaerobic exercise. Aerobic catabolism. Now we're breaking down sugar in the presence of oxygen. Well, lo and behold, look at the byproducts of this. Carbon dioxide, water, and a lot of ATP, no lactic acid at all. Now we're into long distance marathon, even 800 meter, 5,000 meter, that kind of a thing. Okay, now you're in the presence of oxygen. The athlete goes into oxygen debt and now starts breaking down glucose and producing these aspects. And a lot more ATP. All right, so cellular respiration in a nutshell. You'll see here, you'll notice that I'm in the cytoplasm, and then I go into the mitochondria. And let's just break it down here. Glycolysis occurs in the cytoplasm. It is anaerobic. There's no oxygen. It's a very ancient metabolic pathway. Okay? You know, it's not good for producing ATP, but it's good for some organisms because this is all they have. Okay, and you'll see here 1 glucose molecule produces 2 ATP plus 2 pyruvate. Okay, molecules there, and we'll show you those there. All right, and that's, you see, there's your 2 ATP. Okay, there's 1 pyruvate. Another pyruvate right here, and your 2 ATP are produced. Okay, no oxygen. But then you'll see there that these pyruvate molecules, they move into the mitochondria. And that's what's going on here. Once we enter the mitochondria, we're now into what's called the citric acid cycle, or you'll see it called the Krebs cycle. Okay, and it's always denoted by this circular situation here. And then we're just introducing this. We'll hit this later on in a lot more detail. Okay, but once you move into the aerobic, once we've moved into the mitochondria, now the amount of ATP starts to get a little better. Okay, so you'll see there that one of these pyruvate molecules that comes in here and enters the Krebs cycle or citric acid cycle, and in doing so, okay, it converts from a 3-carbon to a 2-carbon. We'll get there, but you'll see it produces another 2 ATP. So there's 2 ATP that was formed here and another 2 that's here. Okay, but what's really going on here now is you'll see that one glucose produces 2 ATP, as we just said, but it also produces 8 hydrogen ions. Okay, and that's what's going on here in the mitochondria, is that it's pumping hydrogen ions out. And so basically what you're doing is you're creating kind of like a battery situation where you have positives on it's more positive on one side of a membrane than it is another. And once you have this differential across a membrane where you have one charge on one side of a membrane and another charge, it's kind of like a waterfall. You're able to do work. And we'll show you this more. But you'll see that that's what's important here, is that from one glucose in the Krebs cycle, you're producing 8 of these hydrogen ions, which are transferred to 6 NAD molecules and 2 FAD molecules. Okay? These will become important in the next step. These NAD and FAD molecules, okay, now you're talking what we call the electron transport chain, also called oxidative phosphorylation. Okay, oxygen phosphorylating, we're making ADP into ATP. That's where the phosphorylation. And it's these guys here that it's the NADs and the FADs. That's where you get all of these ATPs here in the electron transport chain. Okay, and we'll get into that there in more detail there later. And yeah, anywhere from 26 to 28 ATPs, okay, per glucose. So yeah, 2 ATP up here, 2 ATP here, but the big money is these guys here, those 8 hydrogens, okay. That's where the money comes from, because once these guys here are in place, the electron transport chain comes into effect, and then that's where you make the ATPs, okay? Outside in the cytoplasm, inside in the mitochondria, all right? End products there, carbon dioxide, water, ATP, I like that, chemical energy, and heat, okay? Just an overview, which we're going to overview and overview and over and over and over and over again anyway. So here's one of the first overviews. Cellular respiration takes place in 2 stages. There's your glycolysis, which occurs in the cytoplasm. Does not require oxygen to generate ATP. Okay? Krebs cycle, also known as the citric acid cycle, occurs in the mitochondria. Harvests energy-rich electrons and protons, there's your NAD, through a cycle of oxidation reactions. The electrons passed to an electron transport chain to power the production of ATP. And basically, that electron transport chain, it's kind of like hot potato. You have these high-energy electrons that have been released by the FAD and the NAD molecules, okay? And these high energy electrons is kind of like hot potato. You're trying to, they're too hot to handle, so you're bumping them in your hands, and you're trying to siphon off as much energy as you can at a time, and eventually that electron does cool down to the point where there's no more energy to harvest off of it. And we'll show you this there. There's a couple of good slides there towards the end. So the whole idea of oxidation and reduction. So you'll see here that this atom, or whatever it is, you know what I mean, has these 2 electrons, okay? And oxidation is the process of losing those electrons. So here, this species has been oxidized, okay? And where do those 2 electrons go? They went onto this beast here, okay? And you see this guy here in this process gained 2 electrons. That is what being reduced is. That's called reduction, okay? So this guy here that was oxidized, oddly enough, it's called the reducing agent. This one here that was reduced, okay, it gained electrons, it's the oxidizing agent. Clear as mud? I hope so. Okay. There's your mitochondria. Okay. you will see there that there's two membranes. Like these guys here, they actually think that it was a bacteria at one point that the eukaryotic cell engulfed. It has its own DNA, it has its own ATP, it's kind of its own thing. All right, it works for us though. And you'll see there that these folds, okay, these are what we call the cristae. And that inner membrane, you'll see it here, and it follows along here. There's your inner membrane. Well, then there's the outer membrane, okay, which is here. And then you'll see there that there's a space in between the outer and inner membrane. This is called the inner membrane space, okay? It's very important. You remember the whole process of producing hydrogen ions, okay, right in here, right? And remember we produce those hydrogen ions and that are transferred to the NAD and the FAD. Well, where do those hydrogen ions accumulate? They accumulate in the space in here. And when there's so many of those hydrogen ions in the space, then what the mitochondria does is it allows them back in, but now you've got kind of a pump, and they're coming in and making ATP. That's the whole process. All right, we'll get into more detail right in here. So you'll see here, there is your glycolysis. Okay, it occurs outside in the cytosol. Okay, breaks down glucose into pyruvate. Okay, 6 carbon into a 3 carbon. And in that process produces ADP. And as well, we give a couple hydrogens there to this NAD molecule. All right. And now these pyruvate molecules, they now enter the mitochondria and they go into, they get changed into acetyl-CoA, which is now a 2 carbon. 6 carbon to 3 carbon to 2 carbon, which now enters the Krebs cycle. Okay. This is in the presence of oxygen. Okay, we get more ATP. But remember there, it's those hydrogens there that are of the greatest importance, and they get transferred there to the NAD and the FAD, which is then involved in the electron transport chain and siphoning off those hot electrons, and that's where we get all the ATP. Okay? Lots of good stuff there. I love it. There, this video here, watch it from start to finish. It's a great video. It takes you on a tour and explains everything in great detail. So take 10 minutes, 12 minutes, watch the video. It's a great one. Okay. So just going back in here, glycolysis occurs in the cytoplasm. It is the main energy source of prokaryotic organisms that do not have mitochondria. So prokaryotic organisms that don't have mitochondria, this is all they have, glycolysis. It's very ancient, okay? The amount of ATP is not great, but if you're a bacteria, hey, it works for them. It is a sequence of chemical reactions that breaks one glucose molecule, 6 carbon, into 2 pyruvate molecules, 3 carbon. The breaking of bonds is used to form ATP. Okay, this is called a substrate level phosphorylation. Phosphorylation is the process of adding a phosphate to ADP and changing it into ATP, adenosine di to adenosine triphosphate. Also, hydrogen and electrons are donated to NAD to form NADH, 1 NADH for each pyruvate, so 2 total in glycolysis. 2 of these guys here, okay? You're adding a hydrogen. Remember those hydrogens there? You're starting with this and you're finishing with that, and that's what you want. So glycolysis yields only a small amount of ATP. Only 4 ATP are made for each molecule of glucose, but remember, we lose 2 ATP to start the process. So we make 4 ATP, but it costs us 2 to get the process going, so we only make 2 ATP. 1 NADH molecule is produced for every 2 ATP at this stage. Okay, so if 2 ATP are made, 1 NADH is made and saved for later. This is the only way organisms can derive energy from food in the absence of oxygen. all organisms are capable of carrying out glycolysis. This biochemical process was probably one of the earliest to evolve. Okay? There are some questions for you. Have a look at those and let me know if you have any trouble. Okay? Lots of fun. Now, this is kind of a step-by-step. We're kind of, as I said, we're going back talking about this again and again in different ways, because there's different steps to glycolysis and this whole process of metabolism, and we're kind of looking at it at different facets and different angles. Okay, and all I'm looking for you to do is grasp one of those and get it. Okay, because it's not the easiest thing to talk about for sure. All right. So you'll see here that when you're talking about glycolysis, all right, there's 3 phases or events that are going on here. Okay, there's the phase 1, phase 2, and phase 3. So you've got this, what we call phosphorylation. And then we've got splitting. So you'll take your 6-carbon glucose, okay, you phosphorylate it. And you see you added a phosphate and a phosphate. It's still 6-carbon, but you've got a phosphate on either end, okay? And then splitting. And then you cut that 6-carbon into 3 and 3. See what you've got? There's your pyruvate, okay? The beginnings of. So there's your 3-carbon and your phosphate, 3-carbon and your phosphate. Now, you're into the production of NADH and ATP. Now, and remember there, where you really make your money in all of this is the production of hydrogen ions. Because if you make any of these hydrogen ions there, you're able to throw them on a NAD molecule and save those for later for the electron transport chain. Okay? So, glycolysis, as you remember, is anaerobic, very ancient. 10 steps. I'm not asking you there for the 10 steps. Breaks down glucose into 2 pyruvate molecules, occurs in the cytosol, yields 2 ATP glucose molecules, okay, for every NADH. Okay. This is just another shot of the Krebs cycle, okay. Basically, you're going from here to here, right? Okay. And this is just exactly the same thing. Maybe you didn't like this one here. So there's your 6-carbon glucose, okay, and then there's phosphorylation. We phosphorylated there both ends there with phosphate, okay, and then we break it up, we split it, cleave it, and then here at the end of the glycolysis, we now end up with 2 pyruvate molecules, okay. Very important what's going on here and all the different processes. Now, same video, wants you to continue on. Okay, if you stop there previous, go back and watch it, and it continues on with the Krebs cycle, citric acid cycle, as well as the electron transport chain. Okay, so pyruvate oxidation and citric acid of the Krebs cycle. In the presence of oxygen, now remember there, the glycolysis was all outside of the mitochondria, no oxygen. Small amount of ATP, ancient cycle, okay, good for bacteria, not great for long-distance runners, okay? So in the presence of oxygen, the first step of oxidation, oxidative respiration in the mitochondria is the oxidation of that pyruvate, because you remember, that's what we ended up with. That's as far as glycolysis will go. And I don't mean to just jump back here and just, moving all around, but the whole idea. Okay, there's your glycolysis. Little bit of ATP. Now those pyruvate molecules got to come in here, and now we're starting to make some money. Okay, same thing here. Now we need to get these pyruvate into the mitochondria so that we can start making some good ATP. Okay. Now I lost my spot. Of course I did. So, pyruvate molecules. End of glycolysis. Now moving on. Okay. So, these pyruvates still contain considerable stored energy at the end of glycolysis. The first step is to oxidize pyruvate to form acetyl-CoA. Okay, oxidize, what's going to happen there? Okay, it's going to lose some electrons. So, when pyruvate is oxidized, a single of its 3 carbons is cleaved off by the enzyme pyruvate dehydrogenase. This carbon leaves as part of a CO2 molecule. That's it, breathe out. That's where that carbon came from. Okay, a third of the carbon dioxide you breathe out comes from that particular process alone. Okay, seriously, the gas came out of all your cells at one point in time. All that gas was inside your cells. So this carbon leaves as a CO2 molecule. In addition, a hydrogen and electrons are removed from pyruvate and donated to a NAD molecule to form NADH. Aha! The remaining 2 carbon fragments that was left over from the 3 carbon pyruvate now, okay, joined to a cofactor called coenzyme A. Okay, the final compound is called acetyl-CoA. All right, acetyl-coenzyme A. So you'll see outside, inside, and you'll see there that there's 6 carbon glucose, okay, phosphorylated, all right, and then it's cleaved into 3 and 3, and then you'll see there these pyruvates, they come on in. One of those carbons is removed as CO2, okay, a hydrogen and electrons are provided there for an NADH, to a NAD molecule and saved for later, okay, as well as up here. And then now you have this 2 carbon acetyl-CoA that goes along the citric acid cycle, okay, and that's what's going on here. So if oxygen is available, pyruvic acid is used to produce acetyl-CoA. Citric acid to Krebs cycle begins, the electron transport system functions, carbon dioxide and water are formed. Total 34 molecules of ATP are produced per each glucose molecule. Okay, so, and we'll come back to this again. I'm throwing things at you at different levels, and I think that you're seeing how this process works. Okay, and then you'll see all the NAD and FAD molecules that are created here, FADH and NADH. And you remember I was talking about the hot potato? You take those high energy electrons, right, and whoosh, siphon off, whoosh, siphon off, whoosh, siphon off a little bit of energy here and there and here and there until they become inert. But by that point in time, you've created many, many, many ATP. It's interesting there to note that the fate of that acetyl-CoA, remember that 2 carbon, it went from pyruvate molecule, 1 carbon was removed as carbon dioxide, leaving you a 2 carbon acetyl-CoA right in here, right? So the fate of this acetyl-CoA molecule depends on the availability of ATP inside the cell. And if there's a lot of ATP, then what happens there, if the ATP is plentiful, the acetyl-CoA is diverted to fat synthesis for energy storage. But if there is insufficient ATP and the cell needs more ATP to do stuff, then the acetyl-CoA enters the Krebs cycle. Interesting. So if there's no ATP, that acetyl-CoA goes to the Krebs cycle. Okay, mitochondria. If there is plentiful ATP, the acetyl-CoA is diverted for energy storage. So the Krebs cycle is a series of 9 reactions that can be broken down into 3 stages of oxidative respiration. Okay, we're kind of walking over the same carpet, but I think we're doing it in a very, very good way. You see how we're just breaking it down, and we're kind of gaining a little bit of ground every single time. So the 3 stages there, you've got acetyl-CoA enters the cycle and binds to a 4 carbon molecule, forming a 6 carbon molecule. All right, so we've gone from 2 back up to 6 carbon. Again, 2 carbons are removed as CO2 and their electrons donated to a NAD molecule. In addition, an ATP is produced. The 4-carbon molecule is then recycled and more electrons are extracted, forming more NADH molecules and FADH molecules, FADH2 molecules. And that's where the money makers are, right there, right? Okay. So yeah, have a look here. The Krebs cycle involves the production of one FADH2 and 3 NADH molecules for each acetyl-CoA that enters the Krebs cycle. I'll say that again. The Krebs cycle involves the production of one FAD molecule and 3 NAD molecules for each acetyl-CoA that enters the Krebs cycle. Okay, so 4. One of these guys and 3 of these guys for every single one of these guys. Okay, it's a good return. These molecules carry the hydrogens that enter the electron transport chain. As we said, it was the hydrogens that matter. Okay? And you see here for the Krebs cycle or citric acid cycle, there's your 2-carbon, okay, acetyl-CoA. It enters, and it immediately joins there with a 4-carbon, okay, to form a 6-carbon unit, which is then siphoned. Its hydrogens and electrons are siphoned off. There's an ATP. There's a FAD to a FADH2. There's a NAD to a NADH. Again, this is all money, right? And then this whole process there, you'll see the CO2 is removed twice, once here, once here. So now we've reduced that 6-carbon chain down to a 4-carbon, and that 4-carbon is ready to meet up with another 2-carbon acetyl-CoA the next time it makes it round. And that's how the whole process goes. And all of these guys here are saved for later, okay, for the electron transport chain. Just another shot of the same thing. I'm not going to go into too much detail. There's lots of stuff going on here for the Krebs cycle, but I've told you guys there what I'm looking for. There's your pyruvate, 3-carbon down to a 2-carbon, okay, where the 2-carbon acetyl-CoA is met up with a 4-carbon chain, okay, which makes it 6-carbon, and then in the processes there, 2 carbons are removed. Okay, hydrogens and electrons are siphoned off and saved for later, and then eventually a 4-carbon chain makes it around again, which adds up to the 2-carbon from the acetyl-CoA, and the whole process starts again. Okay, this is all just about siphoning off hydrogens. That's all it is in electrons. Same video, have a look at it. It's a good one. Okay, maybe you might want to watch it again. So, essentially, okay, the whole idea, if you just saw the video, the whole point of the Krebs cycle there is to make these hydrogens, okay? And all of these hydrogens there, they get pumped into the space of the mitochondria, the inner membrane and the outer membrane, and that's where they hang out. And then the body makes ATPs by these guys here. They're just being so many of them on this side that they just start filtering through an enzyme there that these hydrogens go through, and in the process an ATP is made. That's how it goes. We're just pumping those hydrogens out, siphoning the energy off of them there, and making ATP. And you'll see the whole idea, remember those NAD molecules and FAD molecules, okay? This is called the electron transport chain, and it's just showing there the flow, okay? And you'll see there that there's all those hydrogens on the outside, and you see how they come in, all right? And the energy for the ATP, you'll see there that these electrons, like the NAD, it enters here, and it's kind of like hot potato. These electrons there, the energy is siphoned off, and it's added to an oxygen molecule, or half of an oxygen. That's why we breathe oxygen, okay? Because what happens here is it's a 2-part. So the NAD and the FAD, and you see FAD doesn't provide as much energy because the electrons enter into the electron transport chain here. The NAD, it provides more energy per electron because it's adding it at an earlier junction. Regardless. So the NADs are the fads. They throw the electrons in here, hot potato, okay, and those electrons there, they get dumped onto the oxygen, which is why we breathe oxygen. That's the whole point, is that oxygen molecule is going to accept those electrons. Okay, but that's only half the battle. All those hydrogens, okay, that were pumped out into the space in between the mitochondria. Remember, there's so many of them in here. Because there's so many of them here, they flow through this enzyme there called ATP synthase. This is the molecule there that makes ATP. Once those hydrogen ions come in, ADP is phosphorylated and ATP is created. Now what happens with that hydrogen? It jumps over here, and oh, you remember there that oxygen there that has an extra electron? Well, that extra electron goes right onto that hydrogen, and this whole process there makes water. So there are the two products of this mechanism. Remember the CO2 that came out here and here? That's the CO2 you're breathing out. And then the whole idea here of the electron transport train, those NAD and FAD molecules that were collected all up here, right? And even up in the glycolysis, right? There was a NAD molecule that was created all the way up there. See? Up here. Another one here. And now the electron transport chain, this mechanism here is happening within the membrane. And you'll see there, these guys are entered, and the energy is siphoned off and deposited here. Those electrons, the hydrogens there, they are outside and they return back. And in this process, they change ATP, ADP into ATP, and they go on to half of an oxygen, which then makes H2O. Heavy stuff, eh? Not so simplified, but I think you guys have a good grasp of it. That video there, very important. Have a look at this. Go back through it again. Okay? So the electron transport chain and chemiosmosis. Mitochondria use this chemiosmosis there to make ATP. That proton pump, okay, pumping those hydrogens out, uses energetic electrons extracted from food molecules to pump protons across the cristae. So that's what's going on here. Those electrons are pumping those hydrogens out, and you're creating a concentration gradient here, which is not natural because you're pumping hydrogens across. So you're going from a low concentration to a high concentration. Okay? You're trying to create a battery. You're trying to create energy, the ability to do work. Okay? As the concentration of protons builds up in the intermembranal space of the mitochondria, the protons re-enter the matrix via ATP synthase channels. Their passage powers the production of ATP from ADP. Okay? Where'd that go? Yep. Good. So there's your NADH and your FADH2 molecules. They transfer their electrons to a series of membrane-associated molecules called the electron transport chain. This is it right here, the inner mitochondrial membrane. So that's where that's happening. So what slide is that? 48. So this process right here of hot potatoing there the electrons to force the protons out, that is happening. I'm just going back into, yes, it's happening right along there. That's where that is happening, that inner membrane, that process of hot potatoing the electron, okay, and forcing the protons out, that's happening right on there. Now, can I find my space? There we go. Good. They transport electrons to each other and act as proton pumps. The last transport protein donates electrons to hydrogen and oxygen, ultimately forming water, as we just spoke about. Okay, just another shot of the same thing. There's your NAD and your FAD. The FAD starts a little later. Okay, I'm not interested in ubiquinone and cytochrome C. I'm not interested in any of these. Okay, just on the mechanism there, okay, of what is exactly happening. And the whole point, these electrons pump those protons out, and once they come back in through ATP synthase, it changes them into, well, it creates ATP, and those hydrogens there, they go on to the water. Okay? Just another shot of the same thing. Feel free to have a look at it. Okay? And again, it's just taking you there. It's adding there the Krebs cycle. There's your acetylcholine. See? So there's your pyruvate there. Okay? It enters in. There's your acetyl-CoA. So it goes from a 3-carbon to a 2-carbon. Okay? And then it meets up with a 4-carbon here. Turns into a 6-carbon. Okay, and you'll see there that NADs and FADs, 2 CO2s are produced as well as 2 ATPs. Okay, but it's these guys here, that's your money maker. And then those NADs and FADs, they go up to the inner mitochondrial membrane, where those electrons are used to pump the protons out. And then those protons, once they come back in, they make ATP. Nice slide, eh? And I apologize on the complexity. We've been kind of walking over the same carpet for the past 49 slides, kind of a thing. But I think there that we've taken it in stages, and hopefully there you guys have a good grasp of what's going on here. Okay? I really do. So, if you're feeling tired, have a look here. A lot of this stuff, it's just based on what you've learned in the past 50 slides. Have a look. If you're feeling tired, this is what you need to make ATP. Niacin, vitamin B3, there's your NADs. If you don't have any of those, you're not making enough ATP. Riboflavin, which is vitamin B2, there's your FADs. Aha, so make sure you're getting niacin and riboflavin. Pantothenic acid, that's B5, there's your coenzyme A. Coenzyme Q, that's electron transport chain protein. Omega-3 fatty acids, proper electron transport chain function as well, that's brain. Okay, your brain is fat. You need fat. Thiamine, vitamin B1, coenzyme involved in production of CO2 during pyruvate oxidation. That should actually mean something now. Magnesium required for ATP function. Fish, chicken, pork, liver, beef, mushrooms, peas, all protein and healthy good stuff. Okay, it's all high in this stuff here. Okay. Need another video? There's 2 for you. Have some fun. Other nutrients that produce ATP, all the macromolecules can be broken down and used to make ATP. Polysaccharides, glucose. Protein, amino acid. Triglycerides, fatty acids, and glycerol backbone, right? 2 different parts. Nucleic acids can be broken down into nucleotides. Okay. Summary of catabolism of proteins and carbohydrates. There's different things there that can be broken down into different ways. And just wanted to show, I'm not going to test you on any of this here, but you'll see there proteins, amino acids, they do enter, okay, at this stage. But you'll see glucose, simple sugars, they enter in at the top of glycolysis. Amino acids do not. They'll enter in at the pyruvate stage. Okay? Same with glycerol. Fatty acids don't enter until the acetyl coenzyme A stage. 2 carbons. Remember that? So you'll see, depending upon the food source, it will enter the metabolism, this whole process at different layers. Okay? Other nutrients there that produce ATP, again, same thing, kind of a different slide. And you'll have a look here. Okay, nucleic acids, you'll see where they end up, where they enter. Proteins can be a number of different stages. Generally, they're your sugars, carbohydrates enter here at the start, and lipids as well. They'll enter in at the acetyl CoA at the 2-carbon stage. Not going to test you on it, just to show you there different types of food sources will enter the chain at different times. One NAD molecule will yield about 3 ATP molecules ultimately, so approximately 34 ATP molecules are made from each glucose. 34 ATP molecules in total, okay, are made from each glucose. Okay. How many ATP molecules would we make from one fatty acid with 18 carbons? 18 carbons, okay, times 3, generally around 114, 115 ATP molecules, okay. 18 carbons divided by 3, 6, away you go. And here are some beautiful questions to keep you occupied and have some fun with, okay. And there's some video as well. Please stop and watch that before continuing. Anaerobic respiration is the absence of oxygen. Organisms must rely on exclusively on glycolysis to produce ATP. Okay, so no oxygen, all you get is glycolysis. Okay, it's not great, but it may be all you have. In a process called fermentation, the hydrogen atoms from the NADH generated by glycolysis are donated to organic molecules, and NAD plus is regenerated. So the whole process there of glycolysis, that H is generated and donated to an organic molecule, leaving this guy here ready to accept another proton. With the recycling of this NAD molecule, glycolysis is allowed to continue. And it just continues and continues. That's your fermentation process. Okay. Bacteria can perform more than a dozen different kinds of fermentation. Eukaryotic cells are capable of only a few types of fermentation. Yeast, single-celled fungi, pyruvate is converted into acetaldehyde, which then accepts a hydrogen from NADH, producing NAD+, and ethanol. That's the process of making alcohol. Okay? In animals such as ourselves, pyruvate accepts a hydrogen atom from NADH, producing NAD+, and lactic acid. Interesting. There's where the lactic acid comes from. Okay? In anaerobic respiration. Okay? So if you're doing running in an anaerobic environment, your muscles will be very sore the next day. More on anaerobic respiration, just the whole idea of what is happening here. Okay, there's your lactic acid fermentation in muscle cells. Okay, again, no oxygen. And this is alcohol fermentation in yeast. Okay, fermentation. So fermentation is, we've used it for thousands of years to make bread, wine, tea, yogurt, sauerkraut, kimchi, yogurt, all these different things. It's all about fermentation. Okay. More questions there for you. Okay. A beautiful summary of cellular respiration detailing all of this. Please have a look at this slide. I hopefully there you are up to date and feel confident with the 3 sections involving cellular respiration. There's your glycolysis, your Krebs cycle, a citric acid cycle, and lastly, the electron transport chain. Carbohydrate storage, okay. You remember your catabolic pathway for energy storage and anabolic pathway for storage, okay. Carbohydrate molecules from food can enter either of these, okay. Breaking down, we can break them down further for energy release, or we can build them up and store them for later. Excess glucose stored as glycogen, primarily by the liver and muscle cells, okay. Glucose can be stored as fat, can also be converted to amino acids, with difficulty, but it can, okay, gluconeogenesis, all right, where we can make sugar from non-sugar sources. Making of glucose, the body must have a constant supply of glucose for fuel. can be made from non-carbohydrate substances. There's your gluconeogenesis, making glucose from non-glucose sources such as protein, important in blood sugar regulation. And a nice summary there. Please have a look at that. There's your fads and your nads. Those are your money makers when you're talking respiration. And here's a beautiful little overview, cellular respiration overview. Total energy somewhere around 34, 36, 38. It can change. Thank you so much, guys. I hope you have a wonderful week. We'll see you there for week 8. Take care and be safe. And if you have any questions, get a hold of me. And don't forget to do all the practice questions in the folder. Thank you and take care.